here now with this part, ultimately, how do we get rid of that nitrogen? We stuck the nitrogen to the liver. The liver's got to get rid of it somehow. And so that's nitrogen excretion via the urea cycle. And once again, we're only talking about, and I'm only going to expect you guys to do the ones that are ureatelic, like, humans, most mammals and sharks. Okay, I won't expect you, I don't know the one for uric acid for alligators and birds. And, well, for fish, they just get rid of the ammonia. They just, and so they just put it out into the water. So, therefore, there is no.
Because of that, well, never mind. I was going to say, because of that, does that mean like eating fish would like screw up our ammonia levels? But if they're excreting it.
They're excreting it. Mm-hmm. So, only if you have a sick fish, I guess. But I doubt that you would have that problem. Usually with eating fish, the problem that you would have would be mercury and other heavy metals because the fish don't have a way to excrete it. So if they imbibe, if they ingest it from the water, from dirty water, then it stays in them just like it does in us, and then you eat it and then it stays in you because we don't have a way to get rid of it either. So that's why there have been cases of people who eat lots and lots of sushi or lots and lots of certain types of fish and then have mercury poisoning.
Is that common in the cultures that eat sushi and things like that often?
I mean, it can, and so that's why they have to be really careful. So more places test for it and things like that.
Like get rid of it, like with intervention since the body can't.
Well, if it's caught early enough, you can get rid of with some chelators or something like that to try to get rid of the mercury. And one of the more recent, I don't want to say famous because I don't know how famous you consider this person is, but the actor Jeremy Piven from Entourage and original Ellen show and stuff like that, he I guess ate lots and lots of sushi and tuna a lot, and so he was starting to get sick, and they couldn't figure out the reason why, and then couldn't find out it's because he had mercury poisoning. And so, we're eating too much. Okay, but yeah, so now we're going to talk about how to get rid of the nitrogen, ultimately from your body. So here's the overall picture, which we've actually have seen before. I just wanted to go over it once again. We have 3 ways. There are 3 ways that nitrogen enters the liver. One is just the amino acids from your proteins ingested, just normal amino acids. Okay. The second way is, all of the tissues, including muscles, but all of the tissues, to do the intercellular trafficking makes glutamine and then comes through. Okay. And then the third way is from muscles under anaerobic conditions that make alanine, and the alanine comes in. Okay, so I'm going to start here once again. So what happens is we have an amino acid. We just use an aminotransferase. Once again, all of the aminotransferases take in this instance, we're going to take, since we're trying to get rid of this nitrogen, it's going to take alpha-ketoglutarate. The alpha-ketoglutarate will take that nitrogen to make glutamate, and then you've made the alpha-keto acid. And then depending upon that alpha-keto acid, it can go on to do other things or be used for other things. Okay. The other way is if it's alanine, alanine is already the amino acid, so we're going to use alanine's aminotransferase. That one's a specific one. For example, if this was serine, it'd be serine aminotransferase for this enzyme right here. But we're going to use alanine aminotransferase. Once again, it's alpha-ketoglutarate, which will make a glutamate in the liver. And then that's pyruvate. Of course, pyruvate can go to make gluconeogenesis. Can go to make glucose. Okay. Or, I guess, under certain conditions, some of the pyruvate is also used for the citric acid cycle and things like that. All right. The third way is we already have glutamine. And so then the glutamine is going to get rid of its nitrogen to make glutamate, which then the glutamate's used for all the aminotransferases. Glutamate is also used for neurotransmitters and things like that. And then this nitrogen, depending upon the species, is either, for those that are like fish, can just, except for sharks, can just eliminate it from its body in the form of ammonia. For those, I was going to say extraterrestrial, terrestrial, that would be interesting. I'm sure I could get lots of comments if I said extraterrestrial. Because a bunch of UFO junkies follow me on YouTube too. But the terrestrial mammals and sharks, they utilize urea. So that's what we're going to cover. And then the birds and the reptiles use uric acid. Okay. Oh, there we go. We can see we've actually already shown this once before, but just to review. And in case I use these terms, you hear those terms. A monotelic just means it can't excrete ammonia. Ureotelic, which is us in sharks, can excrete it as urea. And then birds and reptiles are called uricotelic.
Is uric acid toxic for that? I mean, not like.
I mean, I don't know that offhand, but I wouldn't be eating bird poop if that's what's going to.
But like ammonia is.
Right. I mean,
Uric acid.
I don't know. I mean, it is similar to your bases. And so I don't know if it would be a competitive inhibitor for that or not off the top of my head. Okay. But yes, and this is the reason why birds don't pee, because they just excrete it in their feces as uric acid. Okay. Oh, there we go. So we have ammonia comes into the hepatocyte, in particular to the mitochondria, okay? And so it's converted to urea via the urea cycle. So once again, we have to just think about, we have our cell, which a lot of times I've been drawing it. We have our cell, we have the mitochondria, which how many membranes does the mitochondria have? 2, right? And so we have the outer membrane, the intermembrane space, the inner membrane, and then what's the holy of holies of the mitochondria? The matrix. And so we've been able to try to go through with all these metabolic pathways, place where they're occurring. Glycolysis is in the cytosol. Gluconeogenesis is a little tricky because depending upon when it starts, it can go from the cytosol. Part of it has to be in the matrix of the mitochondria, and then part of it has to be in the cytosol, ultimately. Whereas citric acid cycles where's the citric acid cycle? The matrix, okay? And so, and the one little asterisk is there's one enzyme, and you should know that by the end of the semester, of the citric acid cycle that still has both a matrix side and a side that's stuck in the membrane because it's bifunctional. Part of it does citric acid cycle, part of it does electron transport chain. But the urea cycle occurs within the mitochondria, okay? And so I should say it begins in the mitochondria and then parts of it comes out into the cytosol. So it's bi-compartmental, similar to gluconeogenesis. Yeah.
What molecule is that again? That's in citric acid cycle and.
What molecule? What do you mean? What enzyme?
I mean, yeah, what enzyme?
Succinate dehydrogenase. Okay. So this is the overview, which I didn't bring up my computer. Let me check right here to see the words on it. No, this is the overview, and so this is just like what I would be drawing as that picture, like if I was trying to draw this in my notes, or whenever I've done it in the past in my notes, where we have, I mean, it depends on where we start. If we're starting here with respect to ammonia, it's starting in the mitochondrial matrix, it's got to come out to the cytosol, and it's going to come back to the mitochondrial matrix. So it really is a cycle. Sometimes it's called the Krebs bicycle, because it's also tied into citric acid cycle, which they're only showing it. Remember that it was called that one picture that we showed had what they call the aspartate argininosuccinate shunt or bridge, because this makes oxaloacetate, which of course does. Whoops, I didn't do it in the wrong place. Which does the citric acid cycle. Okay. Give you guys a moment to get caught up, or just to actually start to look over this and start thinking about what all of these are, because I don't remember which one I answered first. And so let me pause this. That's the fun part, digressing. All right, so we are in the urea cycle, and I've separated it into two parts: the part that's occurring primarily in and around the mitochondria, and then the part that's in the cytosol down below. Well, I'm going to say below. Obviously, there's not directionality inside your body. Okay, inside the hepatocyte. Remember, this is a liver cell. All right, so we have one way is you have the liver's amino acids, right, hooking up with this enzyme right here, which is going to take alpha ketoglutarate to make an alpha keto acid, and vice versa. Okay, so what is this compound right here? What is that compound? That's alanine. And where did it come from? The muscle. Okay, So here we're talking about the muscle. I mean, yes, technically the liver also has some alanine itself, but we're saying it's coming from the muscle, okay? And then we also have this right here, and where did it come? What is this compound right here? And that's a nitrogen, kind of got blocked. That's glutamine, and where does it come from? Besides the liver has some of its own glutamine, where else does it come from? The other tissues. And technically the muscle can also, once again, it's not exclusive for alanine, but it can, but yeah, all the other tissues. The term that they like to use is extrahepatic. Okay, so it just means outside the liver. Now let's see. Oh, I did answer both. Okay. So, if we look back here at the alanine, I'm going to go with alanine first, or just the liver's amino acids of its own. Remember, it's hooking up with alpha-ketoglutarate to make the alpha-keto acids. And it also makes this compound. What is this compound right here? That's glutamate. Okay? So that one is glutamate, which I'm just going to put as GLU. Or the letter E, if you wanted to do the one-letter code. On the other end, we have the glutamine that came from the extrahepatic tissues, which both glutamate and glutamine can transverse from the cytosol all the way through to the matrix, that holy of holies of the mitochondria for faith integration. Okay, what enzyme is it that's going to convert a glutamine into glutamate and the ammonia? What's that enzyme right there? What? It's a glutamine becomes glutamate and ammonia. So it's removing the amide nitrogen. Well, it is a type of deaminase, but it's got a very specific name. It's glutaminase. Right, so that one's glutaminase here. I'll just put it. That way he has the prettier. No, let me just double check. Yes. We'll get back to the nitrogen from the glutamine. Now we're focusing on what happens to the glutamate. We've actually covered this already. Does anyone remember what? Because the one caveat is we're going to focus on this one right here, this enzyme. Glutamate can't be used by just a generic aminotransferase because you would never get rid of that nitrogen. It would just be passing it off from the glutamate to whatever other alpha-keto acid. So we have to have a way to get rid of glutamate's amino group, this one. And so, does anyone remember, and it's not shown there, but it does have a very specific cofactor. If I told you the cofactor, you would know the name. What was the name of this enzyme that converts glutamate into alpha-ketoglutarate? So see, this arrow goes like this. It gets rid of that nitrogen. The cofactor that it uses was NADH. It's glutamate dehydrogenase. So glutamate dehydrogenase is the one that removes that nitrogen because you can't use an aminotransferase like we do for all the other 19 occurring. You actually find out there's even more. There's one called ornithine that has an aminotransferase too. But for all the other aminotransferases, because otherwise you just make you can't get rid. It doesn't come off as a nitrogen by itself. It would come off in the form of another amino acid. At the same time, we have oxaloacetate over here becoming aspartate. Okay? And so, this is very important. The alpha-ketoglutarate is also formed. See, there's glutamate. Glutamate becomes alpha-ketoglutarate, and oxaloacetate can become aspartate. What enzyme is that? It's an aminotransferase. What specific aminotransferase is it going to be? What specific aminotransferase is that one?
Aspartate.
It's a aspartate aminotransferase. So I'll go ahead and I'll show that now. So rather than trying to draw it, so glutamate dehydrogenase, aspartate. Remember, the aminotransferases are always named for the amino acid. There may be exceptions, I can't think of it. But the name for the amino acids that's not glutamate. And that ping pong mechanism. We still, technically, this is like leading up to the urea cycle. Just like, or I should say very similar to the step 0 for citric acid cycle, is this enzyme here. We're technically not within the urea cycle itself, but this is prepping the ammonia, because you don't want ammonia to hang around, since it's bad, into what's called carbamoyl phosphate. Okay, so you notice what happens here is you take a bicarbonate, which we actually have quite a bit of bicarbonate. Remember, that's the buffer system that we use in our body for our blood. You get it from it's just carbon dioxide that's been hydrated. Okay, so we have bicarbonate. It requires multiple ATPs in order to make it, to make carbamoyl phosphate. Because technically, when it's just an amide that's by itself, just like an aldehyde that's an end, it's called the formyl group. This is called a carbamoyl group. So it's carbamoyl phosphate. What class or classes of enzymes can do this? Whenever it uses ATP. What class or classes of enzymes utilize ATP? There are kinases and synthetases. Now, is this going to be a kinase or is this a synthetase? This is a synthetase. So if we were going to name it, what would we name it? This is carbamoyl phosphate synthetase. Technically, it's carbamoyl phosphate synthetase 1 because it's the first isomer, but it's the only isomer that you guys have learned so far. See, it's not that bad once you just start to think about and try to understand the logic behind what the enzyme name is and what's really happening. Okay, so I had you guys memorize the 20, and I always stress naturally occurring amino acids that were used to make proteins. I just wanted to point out that these two compounds are both alpha amino acids. They're just not used to make proteins. So we have ornithine, which is named after birds. We have citrulline, which of course is named after citrus fruits. So this is the official first step. This enzyme's name, I get tongue-tied on. Okay, the urea cycle's enzyme names are doozies. I mean, once you see it, it makes perfect logical sense. Okay, so what's covered up, you can't really tell, is this is an inorganic phosphate that's coming off. This is an inorganic phosphate.
So ornithine is just an alpha amino acid that's found in the mitochondrial matrix?
And in the cytosol. Yeah, it travels between the two. Mm-hmm. Yes. Mm-hmm. Yeah, they travel back and forth.
Are they only found in the liver?
I don't know. I'm not offhand. They're just not used to make proteins naturally. Okay, so let's just talk about what's happening here. What functionality, what is actually happening in this reaction? We started off with carbamoyl phosphate and ornithine, and we're making citrulline. But what is this enzyme really doing? It's transferring what?
That amino carboxyl.
Right, and it's called carbamoyl. And so that's why this, if I butcher it, I apologize, because it makes me almost stutter. It's ornithine transcarbamoylase. Transcarbamoylase, okay. But it tells you exactly what it's doing. It's transferring the carbamoyl group to ornithine. I just have problems saying carbamoyl, I think, a lot of times. Transcarbamoylase. And ornithine, of course. So we need citrulline. Now, ornithine and citrulline can come back and forth. Okay. So now we're going to look at what's happening. We're still part in the urea cycle, but now the citrulline can go across and go into the cytosol. All right, so we had step 1, which was the ornithine transcarbamoylase. Citrulline can come on out to the cytosol. Please note, this is the same enzyme, just like when we had, remember aconitase in the citric acid cycle, it did 2 steps. This one has an intermediate. So the first step is ATP is used to activate the intermediate. Okay, so it's called the citrulyl AMP intermediate. And just because, remember, AMP is just a good leaving group. So they've got this color coding. Also, remember, aspartate can go back and forth as well. And we made aspartate from oxaloacetate. So the aspartate comes out, and now the aspartate comes in, and it smacks off the AMP. See this nitrogen? This is great. It's a nucleophile. Boom, smacks that carbon and pops off the AMP. So what class or classes of enzymes can do this? Because ATP is being used, so what are the 2 possibilities there? A kinase or a synthetase. Now, if you think back, what did kinases do? They transfer phosphates typically from one thing to another. What do synthetases do? They're forming something at the expense of an ATP. So is this a kinase or is this a synthetase? It's a synthetase. And what are we synthetasing? Exactly. So, whoops. It is argininosuccinate synthetase. I told you, the name's doozies, but it tells you exactly what it's doing. It is making argininosuccinate at the expense of an ATP.
So it's one enzyme doing this whole.
Right. It has 2 steps, essentially. First, it adds the AMP portion of ATP on it, on the substrate, and then it allows the aspartate to come in and bind and smack off that ATP to make argininosuccinate. Back a moment, get caught up. So then step 3 is interesting. It's got a unique name. So that means we can't just say, it's a transferase or something like that. But what happens here is this enzyme breaks this apart. But I know it's kind of hard to tell. That's why I like the fact that they've color-coded it. The part that it breaks off here is fumarate. So it goes from being a single bond to a double bond, and it's fumarate. Where does fumarate go? Citric acid cycle. So that's one of the reasons why this is also sometimes called the Krebs bicycle or bicycle, because we have aspartate, which came from oxaloacetate. We have a fumarate that's made. Fumarate goes onto the citric acid cycle. So just another way you can think of it, whenever we're trying to get rid of our nitrogen, we're going to ultimately make some energy out of this as well. Okay, but not directly. It's an indirect process. Okay, then we have arginine, because that's arginine. That's fumarate. I guess you could use arginine to, make, if you really needed arginine. Like if you had a diet devoid of arginine, this is one way that you can make arginine. Typically, Western diet, we are not devoid of arginine. However, usually what we're going to do is we're trying to ultimately try to get rid of that nitrogen that's been added on, the nitrogens. And so what happens here is we take arginine. Oh, I forgot, we didn't talk about the name. So this one's got a unique name. I'm sorry. The unique name for this is called argininosuccinase. So we had ornithine transcarbamoylase. We had argininosuccinate synthetase and argininosuccinase. And then step 4, which is the last step, also has a special name. Unfortunately, the urea cycle is short, but it's got multiple special names. And the whole purpose here for it is a water comes in, and the water is going to knock off the urea the urea off of the arginine in order for it to be excreted. And then what's left is ornithine, and ornithine that can travel back up into the mitochondrial matrix in order to repeat this cycle. So this one's got a special name. Does anyone know what the special name is? It's not urease. I know. That's why a lot of things. This is arginase. It's named after arginine. So we had the step 0, which was a transcarbamoyl synthetase 1, technically. Then step 1 was ornithine transcarbamoylase. Step 2 was the argininosuccinate synthetase. Step 3 was argininosuccinase. Step 4 is arginase. And that?
Dr. Jason.
Yes.
So it's just showing that the ornithine is made from the arginase is outside of in the cytosol, and then it travels back into the mitochondrial matrix.
Yeah, in order to repeat the top half. Okay, and then I'm referring to this the fact that some called it the bicycle, or like especially looking at some of the older books, because it is related. The urea cycle and the citric acid cycle are interlinked here, and that via that aspartate argininosuccinate shunt. But it's a mouthful, I know. But now hopefully we can see where we aren't isolated. The metabolic pathways are not isolated one from another. So we have the citric acid cycle siphoning off oxalic acetate briefly to make, using this aspartate alanine transferase to hook up to the urea cycle for argininosuccinate. Same time, or later on, fumarate gets kicked out. Fumarate, of course, then can be made to malate, right? Because we have fumarase. And then the malate can come back in. Because remember, we had to do it that way ultimately, because it's fumarate to oxalic acetate to malate. But remember, we had to use the oxalic acetate can't go directly. I'm sorry, fumarate, malate, oxalic acetate. You understand what I'm saying? It's been a long day already. All right, but so malate can go back and forth, and same thing with fumarate. Okay, so that's the reason why they're all interlinked. So that's this little picture. Are there any questions? Because that's the end of chapter 18 and what we're going to cover for chapter 18. Which means that we're right on schedule once again, which is awesome.